Microplastics are small particles composed of petrochemicals that range in size from 5 millimeters to 1 micrometer. Microplastics are either manufactured to be small, such as microbeads in face washes, or are broken down through physical and chemical weathering over time from larger plastic items, such as straws. Microplastics are found all over the world, from the bottom of the Marianas Trench to the top of the French Alps. Organisms that live in aquatic environments, and especially filter-feeding organisms, are considered especially susceptible to microplastic ingestion. Oysters are a filter-feeding bivalve that are native to South Carolina and are harvested commercially and recreationally. My thesis seeks to investigate the microplastic concentrations of oysters in the state of South Carolina. Today, I'm going to be presenting on the geographic differences among microplastic concentrations in South Carolina oysters. A total of 10 locations were selected through 4 geographic regions. The 10 locations were in regions from North Inlet, Bulls Bay, Folly River, and St. Helena Island, which you can see on the map here. 9 of these locations were from commercial fishing grounds, and the 10th location, North Inlet, was in a National Estuarine Research Reserve that is not open to commercial and recreational harvests and is used as a reference site. 10 microplastics were selected from 3 replicate sites within each location. The oyster tissues were liquefied and poured over a glass fiber filter, where I could then look at the microplastics that could be found within the oysters. Types of microplastics identified included fibers, fragments, suspected tire wear particles, microbeads, and films. A total of 19 microplastics were found in oysters across all locations, or an average of 19 microplastics. While this would mean that each serving of 1 dozen oysters would result in the consumption of 238 microplastic particles. I wanted to examine microplastic concentrations in South Carolina oysters among the different locations. On the first box plot here, you can see microplastics per oyster on the y-axis and the location on the x-axis. It appears that microplastic concentrations per oyster are grouped by location by geographic region. And while they were significantly different among the locations, it was found in a nested ANOVA that geographic region proved to be the attributing factor to differences among the locations. So therefore, the geographic regions were the significant factor in determining microplastic concentrations by location. I would like to use this research in the future to help enact plastic bag bans or other plastic bans throughout the state of South Carolina. Thank you so much.